So, imagine you were trying to get into the most exclusive, high-end VIP nightclub in the world.
Right, the kind where the security is just absolutely impenetrable.
Exactly. You can't just walk in. The bouncers at the door decide exactly who gets to enter the party and who gets tossed out onto the street.
And that is a perfect analogy for your brain, actually.
Yeah, it really is. Because the single greatest bottleneck in modern medicine right now, especially in neuropharmacology, it isn't actually designing the drugs. It is drug delivery.
Right, getting past the bouncer.
Exactly.
Yeah.
I mean, you can engineer the most incredible, highly targeted antibody to clear Alzheimer's plaques or, a brilliant viral vector to fight a brain tumor. But the second you inject it into the bloodstream, it hits a massive brick wall.
The blood-brain barrier.
The blood-brain barrier,
Here.
or the BBB. It is this sovereign border checkpoint. And for you, the listener, we're going on a massive deep dive today into how this elite security team actually gets hired, trained, and put into position before you are even born.
Which is fascinating because for the longest time, we actually didn't know how that happened in humans. The developmental timeline was basically a black box.
Right. And that brings us to our source for today's deep dive. It's a really groundbreaking 2026 paper from the journal Cell, Stem Cell.
Yeah, it's titled Decoding the Spatiotemporal Development of the Blood-Brain Barrier in Human Cortex.
And our mission today is to completely unpack this. We want to understand the exact timeline, the cellular conversations, the secret handshakes that build your BBB from scratch.
And we are really focusing on that crucial window between gestational week 6 and gestational week 21.
So to understand how this barrier is built, let's look at the plumbing. Because the researchers used some pretty wild mapping technologies here, right?
They did, yeah. They combined two incredibly powerful tools. The first is cRNA-seq, which is single-cell RNA sequencing.
Okay, so that's basically looking at the exact genetic recipe that every single individual cell is reading at a given moment.
Exactly. It tells you what the cells are doing, but it doesn't tell you where they are in the tissue because you have to break the tissue apart to sequence it.
Ah, right. You lose the map.
Exactly. So to get the map back, they used MERFISH. That stands for Multiplexed Aerorobust Fluorescence In Situ Hybridization.
Which is a massive mouthful.
It really is. But essentially, MERFISH allows them to computationally map those genetic profiles directly back onto intact slices of the human fetal brain.
Wow, so they can see the exact neighborhood every cell lives in. And when they look at gestational week 6 or GW6, what do they see?
Well, they see that the blood vessels are already there. The initial endothelial cells have grown into the brain. But here is the twist. They don't act like a barrier yet.
Wait, really? So at week 6, they are just like regular leaky pipes?
Yeah, exactly. They're highly permeable conduits. They let plasma and circulating molecules just wash right into the developing neural tissue.
Okay, let's unpack this. If the pipes are already there at week 6, why wait? Like, what exactly happens at GW8 that changes the game?
At GW8, we see this massive sharp shift. A specialized BBB transcriptional signature just kicks in.
It's like the bouncer rulebook is suddenly handed out.
Exactly. Suddenly, these endothelial cells inside the brain start expressing very specific genes. A major one they highlight is MFSD2A.
Oh, right. MFSD2A. That is a critical transporter.
Mm-hmm.
I mean, it usually moves DHA, which is an important fatty acid, into the brain.
It does, yeah. But for the barrier, it does something even more structural. It fundamentally changes the lipids, the fats, in the cell's membrane.
Making it stiffer, right.
Yeah.
So the cell can't form as many transport bubbles or vesicles.
Precisely. It locks down what we call transcytosis. It stops the cells from just swallowing molecules on one side and spitting them out on the other.
That's incredible. And they also start building tight junctions at this point, right?
Yeah, the complexes that physically stitch the cells together so nothing can slip between them.
Okay, so for you listening, I want to do a really quick recap of this first concept here at the 5-minute mark, just to lock it in.
Good idea.
Okay, bullet 1. Your brain gets its first blood vessels at week 6, but they are totally leaky.
Right, no security yet.
And bullet 2. At week 8, the bouncer rulebook is handed out. Genes like MFSD2A turn on, and the barrier is officially open for business.
But that naturally begs the question, who exactly is handing out that rulebook?
Yes, because these blood vessel cells, these endothelial cells, they aren't just deciding to become a barrier all by themselves in a vacuum, right?
No, not at all. They are explicitly instructed by their neighbors. The neural cells, so the neurons and the neural progenitor cells, they are actually acting as the bosses here.
Okay, I love this. So how do the neural cells give the order? Are they like floating a chemical signal over to the blood vessels?
Actually, no. It is much more direct than that. They use a protein pathway called CDH2-CDH2.
Which is a physical connection, right? Like a secret handshake.
Exactly like a secret handshake. CDH2 stands for neural cadherin. And the spatial mapping showed that these proteins have to physically lock together.
So a neural cell extends its CDH2, the endothelial cell extends its CDH2, and they literally grab hands.
Right. And when that homophilic engagement happens, it triggers a massive chain reaction inside the blood vessel cell.
Let's dive into that chain reaction because this is where the molecular biology is just fascinating. It involves beta-catenin, right?
It does. So normally inside the endothelial cell, beta-catenin is constantly being destroyed. There is a destruction complex that just tags it for the garbage bin.
It's like it's constantly being thrown to the biological shredder.
Exactly. But when that CDH2 handshake happens on the outside of the cell, it jams the shredder.
Oh, wow. So the destruction complex shuts down.
Yeah. Beta-catenin survives, it builds up in the cell, and then it travels straight into the nucleus.
And the nucleus is where the DNA is. So the beta-catenin acts as the key to turn on those barrier genes we just talked about, like MFSD2A.
Exactly. It literally flips the switch. And the researchers proved this with a brilliant experiment.
What did they do?
They used a human neurovascular model in the lab, and they genetically knocked down or removed CDH2 just from the neural cells.
So they basically cut off the neural cell's hand so it couldn't do the secret handshake.
Right. And the result was total failure. The beta-catenin got shredded, and the endothelial cells never built the barrier.
That is wild. The whole structural integrity of the brain's blood supply is entirely dependent on getting high-fived by a nerve cell.
It really highlights how interconnected the brain is.
Okay, so we have the pipes, which are the endothelial cells, and we have the instructions via the CDH2 handshake. But a pipe without physical reinforcement is just going to burst.
Right. Especially as the fetal heart starts beating harder and blood pressure goes up.
Right, so enter the support crew, the mural cells.
Specifically, a type of mural cell called a parasite. These cells literally wrap their bodies around the blood vessels to brace them.
They hug the pipes. But how do enough of these parasites actually get to the blood vessels?
Well, this is where the paper discovered something entirely new. The neural progenitor cells, or NPCs, step up to the plate.
Now wait, hold on. I've read a bit about this before, and isn't the classic textbook pathway that the blood vessel cells themselves secrete a protein called PDGFD to attract the parasites?
Yes, you are absolutely right. The PDGFD pathway is the classic mechanism, and it is definitely still happening here.
Okay, so the endothelial cells are yelling out for help using PDGFB. What's the new discovery?
The new discovery is that the neural progenitor cells, especially in a highly active brain region they call the NPZ, are secreting a totally different protein. They secrete PDGFD.
Oh, interesting. So it's a parallel signal, but why do we need both?
Well, the classic PDGFB just recruits existing parasites. But the fetal brain is growing incredibly fast. Just recruiting them isn't enough. You'd run out of parasites.
Ah, I see. So what does the PDGFD from the neural progenitors do?
It acts like a massive chemical fertilizer. It binds to the mural cells and tells them to multiply rapidly.
Wow, it forces them to proliferate, so the expanding blood vessels always have enough support crew.
Exactly. And when they blocked this PDGMD signal in their tests, the mural cells didn't multiply, and the barrier started to break down in those growing brain zones.
That makes so much sense. So let's do another quick mental map for you, the listener, right here at the 10-minute mark.
Let's do it.
Okay, bullet 1. The neural cells physically high-five the blood vessels using CDH2 to give them the barrier blueprints.
And bullet 2. The neural progenitor cells spray chemical fertilizer, which is PDGFD, to rapidly grow the mural cell support crew.
I love how this is all coming together. It's not just a wall. It's a dynamic, negotiated ecosystem.
It really is a symphony of crosstalk. And once all these players are on the board at week 8, the communication just explodes.
It's a two-way street, right? The blood vessels are talking to the mural cells, and mural cells are talking back.
Precisely. The MERFISH data showed that endothelial cells use a signal called DLL4-NOTCH3 to help mature the mural cells.
Right. And NOTCH3 is super important. If that mutates in an adult, it causes terrible vascular diseases like CADASIL.
Exactly. So the blood vessels mature their own support crew. And in return, the mural cells secrete proteins like TGF-beta and ANGPT2 back at the blood vessels.
It's like trying to build a house where the plumbers, the bricklayers, and the electricians all have to constantly text each other every single second.
That is exactly what your brain is doing at 8 weeks old. And they actually found that this texting, this crosstalk, is much stronger in the neuron-enriched zone, the NZ, compared to the progenitor zone.
So as the brain tissue physically matures, the blood vessels mature right alongside it. Spatially, they are locked together.
They are completely linked. But to truly prove how this complex machinery works, scientists always have to look at what happens when the machinery breaks.
Right, because you can't exactly run a genetic knockout experiment on a living human fetus.
No, absolutely not. So they used mouse models. Because the mouse blood-brain barrier develops in a very homologous way to humans.
The timeline is different, but the genetic pathways are basically the same. So what were they looking for in the mice?
They were looking for the master switch, an upstream regulator that kicks off all these transcriptomic changes. And they found a very specific protein called H2AZ.1.
Okay, let's unpack H2AZ.1 because this gets into epigenetics, which is so cool.
It is. H2AZ.1 is a histone variant.
Right, and for the listeners, your DNA is super long, so it's tightly wrapped around these protein spools called histones so it can fit inside the nucleus.
Exactly. And usually when DNA is tightly wrapped around a standard histone, the genes are hidden. They are turned off.
But when the cell swaps out a regular histone for this variant, H2AZ.1, it's like loosening the spool.
Perfect analogy. It poises the chromatin. It unwinds the DNA just enough so that transcription factors can actually get in and read the barrier genes.
So H2AZ.1 physically opens the instruction manual. And here's where it gets really interesting. What happens when they completely deleted H2AZ.1 from the endothelial cells in mice?
It was catastrophic. First, angiogenesis just halted. The blood vessels couldn't grow properly.
And what about the barrier itself?
The barrier signature collapsed. Transporters like MFSD2A totally dropped off, and the vessels started leaking severely.
Now wait, if a blood vessel is leaking, my first thought as a clinician is that the tight junctions failed. The mortar between the bricks broke, and plasma is just flowing between the cells.
And that is exactly what everyone assumes. But the electron microscopy showed the ultimate plot twist.
Oh, I love a biology plot twist. What did they see?
The tight junctions were completely fine. They were intact and physically locked tight.
Wait, really? The paracellular seal was still holding?
Yes. The breakdown wasn't between the cells. The breakdown was straight through the cells.
Oh, transcytosis.
Oh,
Cells
yeah.
went crazy with the transport vesicles.
Exactly. Without H2AZ1, the cells stopped making the lipid transporter MFSD2A.
And because MSD2A wasn't there to stiffen the membrane got floppy again.
Right. And they saw a massive spike in a protein called caveolin-1.
Oh, caveolin-1. That's the protein that literally bends the cell membrane to form those little transport bubbles.
Yes. So the endothelial cells basically reverted to a peripheral leaky state. They were just aggressively gulping up plasma and spitting it directly into the brain.
That is such a critical distinction. So the front door was deadbolted, the tight junctions were fine, but the bouncer left the loading dock wide open for anyone to just walk in.
That is exactly what happened. And they found the final puzzle piece tying this all together. H2AZ.1 actually binds directly alongside our old friend, beta-catenin.
Oh, wow. Full circle back to the CDH2 handshake.
Exactly. The neural cell gives the handshake, beta-catenin survives and goes to the nucleus.
But if H2AZ.1 hasn't unwound the stool, beta-catenin can't actually reach the DNA.
Exactly. You need both the transcellular signal and the epigenetic key to open the barrier program.
That is just phenomenal biology. It requires both the hardware connection and the software update to work.
It really redefines how we view the blood-brain barrier. It's not just a static wall of specialized cells.
No, it's an emergent property. It's a continuous precarious negotiation between multiple cell types.
Exactly.
So what does this all mean for you, the listener? Why should you care about this 2026 fetal brain mapping study?
Well, because understanding how the barrier is built might just be the exact key we need to temporarily take it down.
Exactly. Think about our drug delivery problem from the intro. Right now, to get drugs into the brain, we use really crude methods like blasting the barrier with focused ultrasound or osmotic shock.
Which is incredibly traumatic for the tissue.
Right. But what if we could be elegant about it? If we know the barrier depends on the CDH2 handshake or the H2AZ part 1 epigenetic lock.
We could potentially design drugs that temporarily block that handshake.
Exactly. Imagine if we could engineer a small molecule that tells the BBB to pause just for 2 hours. It induces temporary transcytosis, the loading dock opens up, we flood the brain with life-saving cancer drugs, and then we just let the CDH2 pathway re-engage to close the barrier back up.
It would completely revolutionize neuro-oncology and neurodegenerative treatments.
It really would. We are talking about reverse engineering human development to cure adult diseases.
It's an incredibly exciting frontier.
It truly is. Well, thank you for joining us on this massive deep dive today. And before you go, I want to leave you with one final, slightly provocative thought to just mull over.
Oh, I like these. What is it?
Well, if your brain's neural cells are constantly dictating the behavior of their own local blood supply via these intense physical handshakes,
Yeah.
what if certain adult vascular diseases we thought were just a problem with the blood vessels themselves, are actually failure of the neurons? What if systemic vascular issues actually have a neurological origin?
Oh, that is a whole other rabbit hole.
It is, and we will leave you to explore that question on your own. Keep questioning the incredible machinery inside your head. Catch you next time.